not be a Cape Cod, so enjoy it while you have it.
That's the truth.
Oh, jeez.
It's true.
Sorry.
That's right.
It's
Rob,
just
who
a
were we keeping an eye out for?
Steve Civitelli.
Oh, okay. All right. Thanks.
Leave Alt plus Q button.
34 participants so far. There he is. I got him.
Yep.
Steve Civ.
I'm here. How
Hi.
are you?
Hey, Steve. Rob C. Sean.
Hi, Rob. How are you?
I'm doing very well. How about yourself?
I'm doing well. Thank you.
Awesome. If I could find, there you are. So Steve and the rest of the panel, I'm changing your name to your name plus panel. There we go. All right. 3 minutes and we're recording already. So we got 37 participants.
Now let's wait another minute.
Hey, you got 3 minutes.
Okay.
Give everybody 3 minutes.
I'm going to change my panel name to WISM.
- panel. Make sure you put panel in there because I will.
Left it there. Yeah.
Mm-hmm. All right. There's Chad. There's Chad.
Well, that didn't work out. Uh-oh.
Alrighty then.
I have room for two more.
2 more what?
2 more attendees.
Then what?
I have 46 on the screen now. There's room for 2 more.
Hey, Mike, nice to see you here.
I'm looking for 50 myself.
I don't think I can put 50 on a screen at one time.
I'm at 47.
That's where I am.
All right. 7:31, Ed.
All right. All right, we'll get started. My name's Ed Snyder. Welcome to the COVID vaccine Zoom discussion. This is an informational event sponsored by the Meriden Amateur Radio Club and the Wallingford Amateur Radio Group, which are basically the same group. And I'm president of the group. I'm also a physician and decided that it would be a good idea to have a public education session on the vaccine, since so many of the club members and other people were concerned. So I asked some other folks to join, which I'll introduce as we go forward. But let me turn it over to the administrators for now, who are going to tell us what the ground rules are for handling the Zoom session and the QA period after that.
Okay, thanks, Ed. So the administrators are Rob, K1RCT, co-host me, and Jeff, KC1HDB. So how this is going to work, Jeff is going to be monitoring the technical aspects of our group. If we experience a lot of lag or any lag at all, he may ask us to shut off our video, or he may do it for us. We're asking everybody to remain muted until we get to the question and answer period. How that's going to go. Question and answers will be handled via chat. So if you want to pose a question, let's say you hear something, you want to have a question about it, post in the chat that you have a question, and your questions will be answered in the order they're received in the chat. So only questions that are people that are asking questions of the panel will be handled via chat. So 1, 2, 3, 4, 5, 6. If you're number 2, number 3, whatever, stay muted until I ask for you. I'm going to be monitoring that. Jeff will be monitoring everything else. Jeff, anything to add?
Nothing from my end, but if you're having technical issues, feel free to chat, send me a private chat. I'll be monitoring that as well.
Okay, very good. So I'm going to mute everybody. So everybody on the panel, which is Ed Snyder, Steve Civitelli, and Elsie Mathews, you're going to have to unmute yourself when you go to talk. So I'm going to do that now. Ed, this is back to you.
Okay. Okay, so I am unmuted. If you need to shut off the video, let me know. If we turn off the video, it's so we can get better bandwidth and we won't have falling out and problems. So as I mentioned before, this session is informational. Just a couple of comments before we get started. There's no commercial sponsorship and no affiliation with any organization, academic, commercial, or political. I'm a physician at Yale, New Haven, and a professor of lab medicine at Yale and president of the Meriden Club. And I speak for myself, not on behalf of either Yale or Yale New Haven Hospital. I will be the event moderator. Also on the panel is Ms. Elsie Mathews, who has a master in public health, Whiskey Bravo 1, India, Florida Zulu. She's director of data management for Pfizer, but there is no support was received from Pfizer. She's not speaking on behalf of Pfizer. She just happens to be in our club and work at Pfizer and has information that she can share with us. Lastly is Mr. Steve Civitelli, MPH, Director of Health for the town of Wallingford and President of the Connecticut Association of Directors of Health. So the plan is for each of us to talk about 10 minutes, which will be 30 minutes, cleverly enough, and then open it up for Q&A till about 9 o'clock when we will close it down. So there'll be plenty of time for questions if there are any. Again, this is informational only. It is not our intention to convince you to take or not take the vaccine. And as I mentioned, for Ms. Mathews and myself, the opinions expressed are our own and not that of our employers. Our opinions are, however, based on data, facts, and science, which we will provide. And we went over the ground rules and so forth. So we're going to get started. All right, so I'm going to talk first about what a vaccine is, why do we need one, and is it effective and safe? So a virus is a pathogen that has one purpose, and that purpose is to make more virus. If you're the host and get the virus, you may get a little sick or you may get very sick, depending on what virus it is. The body has several defenses against viruses and other pathogens, which include bacteria, fungi, as well as viruses. White cells are the one source of defense. They're granulocytes, which eat bacteria primarily. And there's another group called lymphocytes, which have multiple subsets. Large one is called B cells, and the other called T cells. The B cells are another form of defense. They make proteins called antibodies. And these antibodies protect us against a whole variety of pathogens. And there are thousands and thousands of antibodies that are already existing floating around. This works well, but sometimes the body needs help. Many of you may have a regimen of exercise, take vitamins, herbs, or supplements to stay healthy. But as the Shingrix commercial says, the coronavirus doesn't care. Sometimes we have to have help to fight a pathogen, and we need antibiotics. Other times we need gamma globulin. And gamma globulin is really a pool of preformed antibody collected from thousands and thousands of blood donors or other individuals that's given to people, and it's preformed antibody for a variety of things. And there's also vaccines. What vaccines do is they stimulate us to make our own antibodies. And that's what they do. They stimulate, it kind of fools the body into simulating an illness so that the body responds as if it were infected, but it doesn't get sick in the process because it's not the virus or the bacteria, whatever that is making you sick, but a piece of it or some form of it, that you don't get ill. So yes, there are side effects with this type of process: redness, fever, soreness at the injection site, but these are signs of inflammation that the vaccine is working. Some people get more of a reaction than others. We'll talk about that in a little bit. Some viruses make us miserable, like the common cold, which can actually be caused by a whole bunch of different viruses. Or there are viruses like COVID, which can make us so miserable it can kill you. 16 million people infected, 300,000 dead is about 1.9%, which means 2 out of every 100 people who contract the virus die. That makes it something to be reckoned with. Vaccines are usually made of live virus that's either attenuated or it's inactivated in some fashion, damaged so that it doesn't act like a virus. The proteins are there to stimulate an immune response, but it's not infectious, or it can be killed. The purpose of it is to stimulate us to fight it as if it were an actual illness without us getting sick. It stimulates your B cells to make antibodies against the virus or bacteria, whatever you're being inoculated with. To fool the body into thinking we're sick, we form these antibodies, so if we see the real virus, the bad one that can actually do harm, we are prepared and inactivated because the body recognizes it as something that it's already seen. How long this immunity lasts can only be determined by testing, doing measurements of antibody levels in actual patients. The flu vaccine is needed yearly because that mutates a lot. Chicken pox you get once, tetanus every 7 to 10 years, and so forth and so on. The S.A.M vaccine, for those of you who remember, for polio was live, but it was attenuated on that sugar cube. The Salk vaccine, which was the first, was killed. Turns out the S.A.M vaccine was more effective. To make a vaccine, they usually grow them on various kinds of animal cells or on eggs. And when they grow them on eggs, you're asked when you get a shot if you're allergic to eggs, because some of those egg proteins are actually in the vaccine, and you don't want to get an allergic reaction if you're very allergic to it. It takes months and months to grow these vaccines, to grow the viruses for these types of vaccines. The COVID vaccine is not made this way. It's made a completely different way using a material known as mRNA. So DNA is a string of genes that serves as the template or the code for literally making us completely, making our bodies and everything that's in it. It directs the manufacture of proteins and everything that causes us to do what we do. DNA, however, does not function in this way by itself. It functions via a messenger that takes the DNA information and converts it into a message that leaves where the DNA lives, which is in the nucleus of the cell, and goes into the remainder of the cell, which is called the cytoplasm. And that messenger then talks to other RNA proteins to build proteins, amino acids to build proteins in the cytoplasm. So there are thousands and thousands of messenger RNAs, or mRNA as it's called. So mRNA stands for messenger RNA. You can think of the DNA as the CEO, the messenger RNA as the COO, and thousands of other RNAs as staff working for the mRNA. It's a little reductio ad absurdum, but I think you get the point. So the messenger RNA is the key that is derived from the DNA generally. That's not the case in the COVID vaccine, and I'll tell you about that in a second. So messenger RNA tells the cells what to make. So in muscle cells, it tells it to make muscle proteins. In the lung, it makes lung tissue, different kinds. And in bone, it makes more bone. How the body turns these messenger RNAs on and off, how it knows what to copy, how do you prevent a muscle cell from making bone or liver proteins, is a very complex process I'm not going into, and it's not well known, and I certainly don't understand it in great detail. COVID virus actually has its own mRNA. In fact, it has no DNA. It's an mRNA virus. There are DNA viruses, but the COVID virus is only made of RNA. So it comes with its own messenger. And what happens when you get sick is the COVID virus gets into a cell, and its messenger RNA kidnaps all the other messengers and everything else inside your cell and forces it to make more of only one thing, which is more COVID virus. So it takes your lung, which is sitting there doing its usual thing, and when the messenger RNA from the COVID virus gets in, it suddenly becomes a COVID-making machine and has nothing more to do with helping your lung. The COVID virus gets into your nose or mouth and then into your lungs, which is why they swab your nose, and attaches. And the reason they do your nose and not the back of your mouth is because the higher concentrations were found in the nose. And then it attaches to your cell. The virus attaches to your cell by the spike protein that's on its surface. You've all seen pictures of the spikes. They look like, the virus looks like a landmine, not a landmine, but a mine floating in the water with the spikes sticking out of it. Only that kind also explodes. This spike protein you can think of as a key, and it's looking for a lock to open the door of your cell. And the lock is on your cell waiting for this key to come towards it, mainly in the COVID virus. Once the COVID spike protein key finds the lock, if you will, on your lung cells, and it finds it on other cells as well, it binds to the cell, and then the COVID virus enters and takes over your cell as the messenger RNA takes over the machinery. While this is going on, inflammation occurs. And this inflammation, if it's in your lung, produces liquid. And if liquid pours into your lung air sacs, you can't breathe because the oxygen can't get across a liquid filled lung. So breathing is nigh impossible. And that's why they have to intubate people to try to force oxygen into the body. And if there's enough liquid, it's not going to work. If it gets in the muscles, you get weak and lethargic. And the inflammation that occurs during this process gives you a fever and a sore throat, and that's much of the symptom complex. Unfortunately, COVID can also cause blood clots because it activates the clotting system. And then if you get a blood clot in your leg, it can travel up to your lung and you get a pulmonary embolism, or it can travel to your brain and you'll get a stroke. And this is part of the reason some people have died of blood clots, and that's the reason it's been stimulated by this virus. The virus has changed over time and mutated, making it much more contagious. The original virus apparently was not as contagious. It mutated sometime around February in the United States, from what I understand, and that made it orders of magnitude more infectious, which is why there's so many people who get this disease. There are apparently also genes in our DNA that either make the virus more virulent or prevent the body from forming substances that will protect against this particular virus. So some of us get flu symptoms depending on which of those genes we have. Others may pass away rapidly because they lack key ones. Which ones they are, what they do, is just beginning to be understood. I just know that there are studies looking at what these other genes may do, and no one knows who have them. You may have them, your wife may not, your children may or may not. That's why it's a crapshoot to know whether if someone gets the virus, they don't get to pick and choose if it's a mild case or not. So we're learning as we're going along with much of this stuff. So to make the vaccine, what Pfizer and Moderna do is they take little pieces of this messenger RNA, either synthetic or little pieces of the messenger RNA. I'm sorry?
10 minutes.
Yeah, that's fine. It's okay. To make snippets of this messenger RNA that tells the cell how to make pieces of spike protein. So what it does is it takes little pieces out of the COVID virus that tells the cell how to make little pieces of spike protein. The spike protein pieces are so fragile that they can't be put into a vaccine. They have to be put, to protect them, they're put into a little glob of fat, which is a micelle. And that little glob of fat with the messenger RNA snippet inside, and that's the reason it has to be frozen at -70 centigrade or -91 Fahrenheit, is to keep the micelles intact. If they let it warm up too long, the micelles would degrade and the messenger RNA would fall apart. So that's why it has to be kept cold. So once the once the vaccine is injected, these micelles with the little snippet of messenger RNA are injected, the micelle dissolves in the cell, and the messenger RNA gets into your cell and makes your cell make these little fragments of the spike protein. When that gets out into the body, your body sees that spike protein as foreign and makes an antibody. And if you make enough of an antibody, it will block any spike protein like that comes through. And that's the key to this whole thing. The spike protein then binds to the antibody to the spike protein binds to that spike, which is basically the key, and prevents it from attaching to the lock. If that is broken, it can't get into the cell, and you may have the virus inside you, but it's not going to get into the cell, so you don't get sick. So what you're doing is, by forming this antibody against pieces of the spike protein, is you're blocking the key so it won't fit into the lock, can't open the door to your cell, and can't get in and infect you. The vaccine does not contain any virus. You are not given any virus particles or pieces of a virus. You cannot get COVID from the vaccine. All it is a little piece of the messenger RNA having you make an antibody against pieces of that spike, but it's enough to damage the key so it won't fit into that lock. The side effects of this are mostly mild. You've heard in the news about a few people that have had more allergic reactions, but it's easier to treat an allergic reaction, even if it's serious, than it is to treat somebody who's drowning in his own secretions because of the inflammation in his lungs. A booster shot is needed for maximum protection, usually 21 days later for the Pfizer vaccine. For the Moderna, it's 28 days. How long does it last? Maybe 8 to 9 months. We don't really know. No one has had the vaccine 8 or 9 months ago. We're still learning as we go. Could there be long-term problems? Yes, there could be. We don't know that either. But you could also get long-term complications from the disease. In 1918, when they had the Spanish flu, millions of people were infected, and it turns out that years later, a large cohort of people who were studied to develop Parkinson's disease were traced back to being infected with the Spanish flu. What's going to happen if you get infected with COVID? No one knows. But you're looking at essentially a possible risk from getting sick, even if it's mild, versus getting a vaccine, which we think is probably safer, although we don't actually know. If the virus mutates, as long as the spike protein is there, your antibody will block that spike protein. And even if it's a mutated virus, you should not get infected if all goes well. The two vaccines, as I mentioned, are similar, and that's how they differ from a regular flu vaccine. That's why they could do it, make it in 2 months, as opposed to when they were talking about not having a vaccine till the end of next year. That was when they were thinking of using it the old way. So I'm going to stop there, and we'll go on now to Elsie Mathews, who's going to talk to us about vaccine production and other aspects. And we'll have questions at the end.
Okay, thank you, Ed. Just making sure I'm unmuted. Can you hear me? Okay, all right,
Yep.
Thank you. Thank you. Good evening, everyone. I want to repeat the disclaimer that this topic is not from Pfizer, but rather public information that I'm sharing with you. And I have several pieces of information to share, and will do my best to give them some connectivity and a reasonable flow. Some of it will repeat a little bit or overlap with what Ed said, but it's always good to hear it again twice, since we didn't compare notes first. Okay, so I'm going to start with a high-level overview of conducting a clinical trial to provide everyone with some background of what was done and how we get this vaccine to people. Then I'll jump a little bit over to manufacturing the vaccine, some details on RNA, and wrap it together with the FDA Advisory Committee output. On average, clinical trials for vaccines typically take years before approval. This COVID-19 vaccine trial data was submitted to the FDA in just 248 days from the time the first patient was dosed to seek emergency use authorization approval. That's only 8 months compared with the average of 8 years. So let's walk through the sequence of events that create a clinical trial. The protocol has to be written, and that's to demonstrate the science. It is designed to show effectiveness against the disease and the safety of the product. A randomized double-blind trial was designed for this COVID vaccine. In this double-blind trial, neither the patients nor the researchers were aware if a patient received placebo or active vaccine. And for this trial, 50% of the patients were randomly assigned to placebo and 50% were randomly assigned to active vaccine. Meanwhile, the investigational product is manufactured, labeled, and ready for shipment for use in the trial. Contracts are created with many research sites globally to ensure we have the proper number of patients that can be included. Then, volunteer patients that meet the inclusion criteria, such as age, demographics, or stable chronic disease, are recruited to enter the study. Researchers administer the vaccine according to the protocol and collect specified data at each patient visit. For this particular COVID clinical trial, many people were needed to manage, collect, review, and analyze the vast amount of data in a short period of time, as more than 40,000 patients were enrolled in this one clinical trial in less than 6 weeks. That's hundreds of thousands of data points needed to be managed very quickly. This is not typical for clinical trials. This was an aberrant situation. A large number of people had to be reassigned from various other projects to just ensure the quality, quantity, and speed of data handling. So now let me shift over to some manufacturing highlights. Manufacturing for vaccines is typically done from specific plants owned by whichever development company is, regardless of their geography. However, in this case, where the world urgently needed distribution of the product, it is critical to have multiple locations across the globe to produce and ship the product. For example, Pfizer has manufacturing is being done in Michigan for the United States, shipments, and in Belgium for the European distributions, in special freezers and shipping containers to get this across those locations. Cryogenic conditions are required for shipping and storage of this formulation for the COVID-19 vaccine. The mRNA vaccines must be kept at an ultra frosty temperature. Pfizer at -70 degrees Celsius and the Moderna vaccine at -25 degrees Celsius. They both require special storage freezers and shipping containers. We don't normally store vaccines at that cold of a temperature, so definitely a new challenge. And as Ed mentioned, both the Pfizer and Moderna vaccines are a new type of vaccine. They're messenger RNA based for which the low temperatures are necessary to really keep those fragile components from breaking down. Let me give a little more description about RNA that may hit home with folks. I want to repeat the point that freezing RNA-based vaccines keeps their fragile components from breaking down. That's the cold requirement starts with the difference in chemistry between RNA and DNA. As Ed mentioned a bit of this before, but I'll give you a little snapshot of my thoughts. RNA is much less stable than DNA, and that's due to the difference in the particular sugars that make up the respective molecules' backbones. When your cells have a job to do, like fight infection, they usually need to call in some proteins to service. The cells have to make new batches each time. The recipe for making proteins is typically stored in DNA. So rather than risking damaging your DNA recipes, cells instead make RNA copies, and those copies are read by cellular machinery. Like a Mission Impossible message that self-destructs once it has been played, many RNAs are quickly degraded once read as well. There are a host of enzymes dedicated to RNA's destruction floating around in your cells. Sticking RNA-based vaccines in the superfreezer prevents the enzymes from tearing apart the RNA and rendering the vaccine inert. So now let me try and connect this information together and briefly summarize what happened at the FDA Advisory Committee meeting last week when they shared their analysis and opinion on the Pfizer vaccine. And as Ed mentioned, they're doing the Moderna vaccine this week. So last week, it was expressed at the FDA Advisory Committee meeting that it's a top priority for the vaccine to be consistently manufactured at highest quality standards. The current Pfizer dose, as Ed mentioned as well, is 30 micrograms at 2 doses, 21 days apart, where the Moderna dose is really 100 micrograms of 2 doses, 28 days apart. The vaccine, in either case, induces neutralizing antibodies, and that's how it works. So at the same meeting, there was communication that a data monitoring committee met weekly to review unblinded safety data from that clinical trial. They did not identify any major safety concerns with the vaccine. There were minimal side effects, such as redness, pain at the injection site, swelling, and probably the most common side effects were fatigue and headache, which are very common, but still weren't seen that often. The committee asked if there was any evidence of infection 7 days after that second dose. It was something they were looking at to see that you're really protected. And the answer was, yes, there was some infection, but only 8 patients of more than 18,000 patients in the active group displayed that, while 182 patients of the 18,000 plus patients in the placebo group had some evidence of infection. It was concluded that this was a rigorous and robust trial, including nearly 44,000 patients with consistent 95% efficacy across age, gender, race, and ethnicity, demographics, plus various comorbidities. The vaccine was well tolerated across all populations with no serious safety concerns. Therefore, it was submitted not only to the FDA, but also to multiple other countries' health agencies simultaneously. And those approvals are happening now as well, so distribution can occur around the globe. In summary, the clinical trial demonstrated high efficacy and effectiveness with a strong safety profile. However, there is still a need to collect more data for certain categories, and that's typically where people have questions. Some examples include pediatrics, children under 16 years old, pregnant women, and protection duration. An important note on at least this clinical trial, the long-term data will be collected by following the nearly 44,000 patients that volunteered for 2 years post their dosing. So in 2 years, we will still be collecting data. So I think I'm going to stop there and turn it to Ed to introduce our next speaker.
Okay, thank you. Our next speaker is Steve Civitelli, who is the Director of Public Health in Wallingford, and Department of Wallingford, and he will talk to us about distribution and other aspects of public health. Steve?
Good evening, Elsie. Good evening, everyone. Thank you for having me here tonight. I'll try and keep it as brief as possible. I think up until this point, we'd all acknowledge that something that we're very familiar with, we have been working more with non-pharmaceutical interventions, right? So our social distancing, our hand washing, isolation and quarantine. So essentially, as a population, we have been fighting with one hand behind our back. Now with the introduction of a vaccine, we have the ability to have a pharmaceutical intervention to help provide herd immunity through means other than natural infection. So at this point in time, from the public health aspect, I think as Ed did a very nice job of talking about how the virus functions, and Elsie talking more about the data sets, at this point in time, we're looking more about the logistics of it and how we ration a vaccine and provide it to a population in Connecticut of 3.5 million people in a way that meets health equity, protects healthcare workers, et cetera. So I could kind of go into a little bit as to how that's taking shape right now. I'm sure many of you have heard in the news that's pretty well known that they're doing this in phases, phase 1A, phase 1B, phase 1C. And then they go to phase 2, et cetera. What all that really means is CDC has a panel of experts that look at over time to see who's in a situation where there's a ration, a limited portion of vaccine available, who are the most at risk? And I think we'd all acknowledge at this point in time, most of the fatalities are, at least in Connecticut, are being found in those who live in long-term care facilities. So in that phase 1A group in Connecticut, from the governor's office down through the Department of Public Health with CDC documentation, in Connecticut, they are looking at vaccinating long-term care facility residents and employees, as well as hospital workers, paid and unpaid, in that very early on group. My understanding is that the first, I'll give you some numbers. So the first allocation of vaccine that was provided to Connecticut was a little over 31,000 doses. It was roughly a 50-50 split between hospitals and long-term care facilities. Many right now of the long-term care facilities are being actually vaccinated by CVS and Walgreens through a federal contract, which is something we've never done before, in that we're using private companies to actually vaccinate these long-term care facilities paid by the federal government, which is very unique compared to some of the other planning measures that have been taken place in the past or have been looked at. What we're also seeing now is something that the hospitals don't typically do, is vaccinate their own workforce. Hospitals aren't mass vaccinators. They're working through this just like the rest of us at this point. Obviously, they were given the Pfizer vaccine, which was approved through the emergency authorization from FDA first. Moderna is expected to get their emergency authorization sometime early next week. So then we'll have two different types of vaccine available to start to really provide to hopefully get herd immunity through vaccinations. And from the timeline aspect, I know a lot of people have a lot of questions regarding that. And here's what I could say as of this point. We're looking at phase 1A, phase 1B being hospital workers, critical workforce, those who are identified at high risk in congregate living facilities. I think the intent is to have those populations vaccinated over the next 2 to 3 months. I think a lot of the timelines are going to depend on the availability of vaccine. And the probably more underrated part of it is the ability to logistically provide or administer the vaccine to individuals. We've never tried to administer vaccine in this fashion this quickly. So typically where you get your vaccinations is that your normal medical provider, maybe you get your flu shot at CVS, but never in this fashion. So this is something that I think it was described to me as we're building the plane as we're flying it, but to a certain extent, we're following multiple layers of guidance. It's CDC guidance. We're looking at state of Connecticut guidance from the Department of Public Health. You have emergency management department for the state, as well as local towns, as well as local health departments working together to try and come up with plans to mass vaccinate. I could speak a little bit more about locally. I know a lot of you are from the Meriden, Wallingford, Southington area. You are all served by local health departments that will probably be vaccinating the critical workforce over the next few weeks. I know that local health departments have not yet received vaccine. It appears that may happen at some point at the end of this month or early in January. And I believe that over the next few months, once that critical workforce and that phase 1 allocation plan devised by CDC takes place, what most people want to know is, when is it going to be available for general public? And I think a lot of that is going to be dependent upon, again, as I mentioned earlier, how quickly can we vaccinate people? What's the actual supply chain, how that looks, as well as what's the vaccine hesitancy. Dependent on what's available and what people will actually utilize will make a big difference on what's available to the general public, maybe sooner or later. Dependent will drive that. So I think at this point, I'm sure that there's going to be a lot of questions. I think my predecessors handled a lot of the questions, or at least made some points that I was going to, so I won't be redundant. I will say that, again, we had 31,000 doses in the state of Connecticut this week. Next week, I believe we're looking at 100,000 doses coming to the state of Connecticut. 17,000 will be allocated to long-term care facilities, 43,000 to hospitals, and roughly 50,000 to a network of clinics. potentially local health departments, et cetera. So I guess I'll stop there and open it up to any questions that are, kick it back to Ed.
Okay, thank you. All right, so administrators, how are we going to handle the Q&A session here?
Okay, I got that, Ed. I've already got several that are posted in the chat. Let's see, we're going to start with the number one. KC1HDB, that's Jeff Martin. Question: I heard there's possibly a new strain of COVID-19 in the UK. If there is one, does the process of making a new vaccine take just as much work or time? Or once you make the first, is the new strain vaccine much easier?
Well, the way we'll do this is I'll take the question and pass it on to anyone else on the panel. And if I take it, anyone else on the panel can just jump in. It's hard for me to answer that, but I would think once a new virus is or a mutant form is identified and can be sequenced to find out what makes it up, if it turns out that the spike protein is similar to what's already there, as I mentioned when I talked, my belief is that the vaccine should be effective against that new virus because it's going against the spike protein. And if it can't get into your cell, presumably it's not going to be able to do that much damage. So I would say they don't have to start over from scratch. When a new virus comes along, brand new, that's unrelated or different enough that they have to make a new virus, I would think a new vaccine, I would think it would only take relatively a much shorter period of time, maybe 2 or 3 months to get something going, but then it takes for the distribution. It's still going to take months and months to get it going. So, and then to vaccinate everyone. It all depends on how infectious it is. Elsie, do you want to join in on that?
Yeah, I'd have to second what you're saying there, Ed. For a new one, that's the exception. It would take a little extra work just to make sure that we're meeting all those needs and understanding what the code is of that specific change. However, for something that's just a minor tweak, potentially, that would make a difference that we still would have to hope that the current vaccine would work for it and just maybe not quite as well. Maybe it's only 70% effective instead of 95%. Depends on what that mutation is. It's really hard to say.
Right. It may be, I mean, maybe a small change molecularly, but it may be a gigantic change clinically. For example, if it may mutate to something that's not infectious anymore,
Right.
That would be wonderful, or it could be even more infectious. By the way, one thing that Steve was, and I talked about mentioning was that even if you get this vaccine, it does not mean that you can throw your masks away, you don't have to socially distance, or you can throw your soap away. You still have to maintain that because, as I mentioned, you may be protected, but you still may be infectious to the people around you. So we're going to be with masks and social distancing for a while, at least through the bulk of 2021, I would think.
Yeah, this is just an extra tool in your tool chest, the masks, the hand washing and social distancing and the vaccine.
Steve, feel free to jump in also anytime you need to. Okay, Rob, you have another
Thanks.
one?
Yep. So Jeff, does that answer your question? KC1HDB.
Yeah, that was helpful, Ed and Elsie. Thank you.
Okay, so we'll move on. This is from Whiskey Oscar 4 X-ray, and I don't have a first. Oh, it's Jim R. Jim R. His question is the only vector through mouth or nose? What other mucous membranes?
Well, pretty much any mucous membrane will do. They're now passing out eye goggles to people working in hospitals. You're absolutely right. It's mostly in the nose, but there are people who can get infected through the eyes or through, I don't know about open cuts or wounds in your hand, but other mucous membranes that may be exposed, if the virus can get in, it certainly can get to the cells that it needs to. So yes, you're right. And that's why those face masks, those plastic clear shields are used, but they're always used over a mask. It's never just the shield without the mask. The mask is your best protection. And for those of you who might think that there's not a lot of data, there have been studies in towns where masks were used and other towns in the same state where masks were not used. I'm particularly thinking of Kansas, although there were others. those towns that used masks had a much lower rate of infection than those that didn't. So as dumb as it seems and as annoying as they are, they actually do save lives. That's my thought. Steve, you want to comment on that?
I think at this point, Ed, well taken, as well as actually goggles are a good option as well, because you have that enclosed seal around your eyes is something that we're seeing more in the healthcare facilities. But yes, to your point, masking is key. The mucous membranes, again, being the eyes, nose, and mouth, would be the big three. We haven't seen transdermal transmission at this point. I think the point there for washing your hands is that if you have virus particles or virus on your hands and then you're touching your face, which I know I do a million times a day, you're trying to, it's the human behaviors that help spread this more than anything. So I think to your point, those are the big three.
Okay, next.
Okay, thanks. This is from me. The question is, any other reactions to the COVID-19 vaccine other than allergic reactions? That's all I've heard about. Are there any other reactions that anyone else has heard about?
Well, there was the ones you've heard about, the anaphylactic reactions, or anaphylactoid, which is really a euphemism. Anaphylactic reactions are the kind of reactions that you see in people who get bee stings or get a penicillin shot. What happens is the offending agent, whether it's peanut protein or egg protein or penicillin or latex, it stimulates a whole series of other proteins. It was one group called the complement system, which results in blood vessels dilating. And when your blood vessels dilate, your blood pressure falls and your heart rate speeds up. So you wind up getting hypotensive and you're tachycardic, as they say, your heart rate speeds up. And if your blood pressure drops enough, you'll pass out. So you see people who are fainting, they lose consciousness. They may get severe swelling around their throat so that they can't breathe. That's the kind of anaphylactic reaction. The treatment for that, many people carry around with them, are Epipens. It gives epinephrine, which tends to get this process under control. Those are the ones that those two nurses in England had, and I believe they had one or two in the United States already. Elsie talked about 44,000 people being treated, which is an actual drop in the bucket compared to the 310 million people who were trying to vaccinate potentially, not to mention the billions around the world. So these clinical trials, even though they're very large numbers, they pale in comparison to what happens when it gets out to the population in general. The reason I think people are moving so quickly is there's been so little in the way of adverse reactions seen that there's a lot of confidence. And look, I'm not the Fauci of Wallingford. I just rely on people that I listen to, that I trust, like Fauci, like Dr. Fauci, like Peter Marks, who's the head of the FDA, who was in charge of the vaccine section. He was a physician at Yale. We're actually good friends. I do know him. He's like Santa. I know him. I trust him. And I said, Steve, how do I know if this vaccine is any good if it gets out? He said, Ed, if I'm still working at the FDA when this vaccine is released, it's okay. Because he would have walked away from it if it wasn't. And I always remember that. So I trust him. Other things, they did talk about a couple of infections that I heard today. Somebody, or Elsie mentioned that there were infections of, I'm not sure what kind of infections, but you wonder if this thing affects spike proteins. Might it affect something else we need that's good for us that we're blocking as well? Doesn't appear to be the case, because there were more infections in the placebo group that didn't get the vaccine than it was in the vaccine group. So the lights look green, but there could be other things. We just have to keep an eye out.
And then and just to add to what Ed was saying, Rob, I think the other thing is a lot of the mild reactions to your typical injection type responses, redness at the site or soreness there, fatigue, headaches, those type things are very common with any vaccine. It's not really related to the COVID-19.
Okay, so what I'm hearing is essentially no, other than normal stuff that we get from any vaccine that we would get. Would that be accurate?
It's very, very rare.
Yep.
Yes.
Okay, thank you for the answer. So the next question is from Jim N1ZN, Jim Savage. Who determines the scope of the trial? Is the developer of the vaccine, is it the developer of the vaccine, or is it dictated by FDA or some other agency? So who determines the scope of the trial?
The developer proposes it, and the health agency, depending on if it's the US, it would be the FDA, or if it's Europe, the EMA, or Japan, PDMA, that they will actually either question it or agree with it. So it's the developer that proposes it, and then it needs agreement from the agencies.
You know, and that is a critically important point. If the FDA does not approve the vaccine, it doesn't get sold. And all the billions of dollars of research effort put into this is for naught. The companies know that. So they don't do their study and then present it to the FDA and expect them to nod and say, good job, guys, what's for lunch? They have a very, very rigorous review process. They had the FDA advisory panel is independent of the FDA. And that was a concern that things were not being, the FDA was not being forced to move faster than it should. And that's what Peter Marks said when he said, if I'm still here, that you don't have to worry about the vaccine. It's proposed and worked with the FDA so that when they do present it, the FDA nods knowingly, yes, this is what we wanted to see.
Okay, so Jim, does that answer your question? N1Z on Jim. Okay, so maybe he moved on. This is from Whiskey Oscar for X-Ray Jim R. I've just had the first of 2 shingles vaccinations. The next is due in 2 to 6 months. Should I wait until after I have the COVID-19 vaccination before taking the second shingles vaccination?
My understanding, that's an excellent question, is that no, I wouldn't take a shingles vaccination and then the next day get the COVID vaccination. You can get it in a couple of weeks. After one, you can get the other. They don't cross-react as far as I know. I don't think there's any problem. Steve, do you have any more information on that?
I think at this point, based on the knowledge we have of the virus and trying to go through some of the safety profiles, contraindications, et cetera, we are at this point, we're still recommending you speak to your healthcare provider.
Right.
Is going to be our default to that at this point. And again, because this is through an emergency authorization, I often remind people that the phase 1, 2, and 3 trials are all still simultaneously going on. So the studies aren't over, right? So they're continuously being done. They're still following the phase 1 folks. They're still following the phase 2 folks, phase 3. So I have a feeling that as more data becomes available in the longer term, as those trials become longer term, we'll have better answers to a lot of these questions. But I think from the public health aspect, when people ask me that, we're going to default back to the medical provider.
Right.
Okay, moving on. Pardon me. This again from Jim Savage. Are there any quality checks on the vaccine after it has been shipped to a location? An example, is the vaccine tested to see that it has not been degraded after having left the manufacturing facility and being stored at a location for a period of time?
The actual vaccine shipments are temperature monitored the whole time. And if that temperature stays there's a sensor in each box. If those stay at that temperature, there's no QC done, shall we say, once it arrives, just for the temperature is the only thing we're monitoring right now. And that is an indicator based on the work that's been done.
And a lot of it from this point, from the public health aspect, is, as was alluded to, is a cold chain management. And that once we receive it from local health or the hospital receive it, they'll do their checks, make sure that there's temperature logs are taken, that's monitored. Obviously we know that there's a temperature range that varies based on the type of vaccine that it is. So we have to log those. And then there's a specific lot number that's associated with each of the vials. So when we have our barcode scanner, we will scan those 10 dose vials. And we'll be able to do a look back in case there's a recall or anything of that nature as well.
Okay, perfect. Thanks. Jimmy, you still here? Does that answer your question? Nothing heard. We'll move on. This from Len Gersha. If I had been hospitalized in the early days of COVID, should I still get vaccinated?
Yes. That question was asked, and a lot of the information I have is from the town hall meetings that I have at Bridgeport and at Yale, New Haven. If you have had COVID and you have recovered, you still should be vaccinated because you can get it again, and it's appropriate to do that. So that's the information I get from Tom Balsazak and from other individuals, Rick Martinello, who's the head of ID at Yale. Yes, the answer is that's what I would recommend. They would recommend.
Thank you.
Okay. Moving on. This from WA1EXA Mark P. Do antihistamine medications have any effects on COVID? I saw a paper that suggests that HIV patients have some protection from the drug.
What kind of patients?
HIV patients.
HIV patients have. Well, I'm not sure how that relates to the antihistamine part.
It's not antihistamine. My typing. It changed.
Antivirals.
I wrote it as antivirals.
Oh,
I'm not sure what it depends what the antiviral agent is against. There's been some comments that there's been very little flu seen in Connecticut, and they're wondering whether, wearing a mask and protecting yourself from COVID also results in protecting from the flu, which it may well be the case. Whether taking other kinds of medication to protect against one kind of illness or viral condition versus another, I haven't heard anyone recommending that. If you want to get protected against the COVID virus, you should take the COVID vaccine. One of the ones is what my recommendation would be, as my knowledge.
And again, from the efficacy aspect, we're seeing right now 94% roughly for the Moderna, 95% for Pfizer. Just to provide a frame of reference, the measles vaccine is 93% in terms of efficacy. So we know that if it's in that ballpark, that's a very effective vaccine.
Mark, does that answer your question?
Yeah.
Yes.
Okay.
Thanks.
Yeah, I saw that. Thank you. Okay, so this next one from N1API Al Kaiser. How long before they feel the COVID curve will flatten out? And will we be able to be notified that a vaccine is available for us?
Well, let me just comment on the flattening part, and then I'll let others chime in. Actually, the best way we have of predicting when the curve is going to flatten is by studying wastewater. The water that people, when you flush your toilets, that water is sampled. And if you measure the amount of messenger RNA in the wastewater, that will predict how many people are going to come down with COVID viremia in about a week or two. The reason is apparently when you get infected, you eliminate the virus in the stool along with the other things you're eliminating. And by the time, however, it takes to get into your lung, you get sick and sick enough to come to the hospital. About another week or so has passed. So by looking at the stool material, the sewage, which is done, I think, on a daily basis now all over the country, serendipitously, who would have known to look there? You can predict, and it predicted last week that there would be a flattening of the curve in Connecticut, and absolutely it happened. We are flat in Connecticut. We've had about the same number. It's still high, but it's not increasing. And they think that there may not have been this Thanksgiving spike that they were thinking of seeing. If we can get through the Christmas period similarly, that would be wonderful. But it's going to, that's the way we know the curve is flattening ahead of time, is by looking at the wastewater analysis. I did see on television that last week that it had gone from 100 mRNA per whatever volume to 600 in Boston. And it was frightening because they had no idea where they were going to put that many patients if 6 times the number of patients got sick. I don't know what the numbers are in Boston. Maybe Steve does, but that's the way it would be looking for now. And I'll let the others talk about the vaccine availability.
Steve, I'm
I
going to turn
think-
it to you so it's distribution locally.
Yeah, sure, not a problem. So I guess I would say in terms of flattening the curve, to Ed's point, you may see it's somewhat flattening. I think what we'll see is intermittent surges over the next several months. I think our hope is in public health is that if we mimic what we had last summer, and that the infection rate is lower in the summer, and over the next several months we're continuously vaccinating, next fall we won't see the dramatic rise that we saw this year. I think that's the ultimate hope that we all have. And the more vaccines that make it out to the street will be more effective in actually doing that. I think, again, from in listening to some of the governor's discussions, general public, they are estimating at some point in May or June. And I think at that point, May or June, you would probably go to CVS, to a Walgreens. You may go to your local health department. You may go to your physician. I don't think it's going to be a one. I think it's going to be multi-outlets for this. I don't think we could do it in a way with 3.5 million people in the state of Connecticut where you have one person doing everything. It's going to have to be done as a team. And I think that'll be the most effective way to get vaccine out as fast as possible.
That's a really good point, Steve. I think my company would agree with you. They've taken a proactive stance in providing options for people to try to stay healthy. And I think that will extend to vaccinations. So the next question, so Al, N1 API, Al Kaiser, does that answer your question? Okay, I'll assume so. The next question from Kristen Olson, KC1ISI. How long after getting the second dose does an individual get to that 95% effective rate in terms of days or weeks?
Some of the first data showed within a week after the second dose. I don't know, Ed or Steve, if you've had more information from the public.
I don't have anything specific, but my understanding was there was some evidence that even after the first dose, about a week to 10 days afterwards, there were some signs that you were starting to be protected. But to be fully protected, you do need that booster. And if you get the first shot is the Pfizer, the second one needs to be Pfizer as well. There's been no studies on Pfizer Moderna mixing. If you get one, you don't need to get the other. I think going back to Al's question, I think of having Moderna virus vaccine available as well will also help to shorten the time period to get everyone vaccinated that needs to be. Probably you'll have to make an appointment to go to CVS unless you want to stand on those lines that you see that look horrific of people standing there waiting to get tested. You go in at your appointed time. There is one question. I don't know if there's any nurses. There must be at least one nurse on this group of 55 or 56 people. Every shot that I have seen on television, they put the needle in the arm and they push the plunger in. I was trained to pull back to make sure you're not injecting into a blood vessel. I don't see that by anybody doing that. And I'm wondering if you don't have to worry. There's obviously not a lot of big blood vessels in the arm like that, but are they doing it the right way? And I'm living in the past, or I don't know if there's any other people who can answer that question.
So I don't know if I can answer that question, but I give my wife a B12 shot and they taught me the same thing. They taught me that you got to pull back to make sure you're not in a vein, and then you can give the injection.
Ed, we've done the same thing in EMS for years. I don't know. Maybe there's some new delivery guidance with this vaccine.
What do you mean? You pull back all the time?
Yes, sir. Every injection we use.
You see, I know stuff. I know stuff.
A lot more than I do, that's for sure.
All
Ed.
right.
Well, I would hope that you would know stuff, Ed.
Yeah, well, you never know. All right, what's next?
Okay, let's see. Where was I at?
Let me throw one in before I forget.
Go
The
ahead.
question about should pregnant women get the vaccine? Right now, there's one organization of the Maternal Fetal Medicine Society recommends women get it. What I don't know is in what trimester they're in. I don't have enough data to know about that, and obviously check with your provider. I would think it would probably be a lot safer in the third trimester after all the organs are formed and the 10 hands and the 10 toes and fingers are there, and you've got a nose where it's supposed to be, as opposed to doing it in the first trimester. Or if you don't even know if you're pregnant, if you take a dipstick and it's positive and you just get the vaccine, I would think it's probably wiser. But it wasn't tested on pregnant women. Does anyone else have any other comments on that, Elsie or? So I would talk with your provider first about that. Elsie or Steve?
Yeah, completely agree with what you've said, Ed, because there wasn't any data to support it one way or the other.
Yeah. You wouldn't do a clinical trial on pregnant women right out of the blocks.
Not unless the drug or vaccine is for pregnancy issues.
Right. Exactly. Steve, any comments from the health department?
No, I'll defer to you both on that. I did, I think it's definitely up for interpretation as the population starts to receive more vaccine. I think we'll learn more, and as the trials expand, we'll definitely learn more.
Yep.
Okay. All right, Rob.
Okay, thanks. I've got maybe 5 or 6 more questions or something like that. Next one from Ted KC1DOY. How long after thawing can the vaccine stay effective? Will it break down quickly?
Well, from what I've heard for the Pfizer vaccine, it's about 5 days at refrigerator temperatures, and Moderna is a bit longer at refrigerator temperatures. And Steve, you may have more instruction based on what you've been delivered.
Yeah, so I know a little bit more about the Moderna because that's what we're expecting. So as of right now, the Moderna vaccine could be held at refrigeration temperatures. It could be frozen for 6 months, refrigerator temperatures for 30 days, 12 hours at room temperature in the vial, and then 6 hours once the vial is punctured for Moderna. The vials have 10 shots per vial. So again, when we open up that first vial, we're going to try and get 10 people, so we're not wasting any vaccine. So that's the other part of this, is to schedule appropriately where we don't, have 8 people coming to the health department for shots and we're wasting 2 shots within that allotted time period. So a lot of the logistics of this is going to be quite interesting. Again, when you're considering it's a 2-dose vaccine as well. I know that the Johnson & Johnson vaccine is going for their EA at some point in January. That is a 1-shot vaccine, and AstraZeneca as well is also out there. So you may have 3 or 4 different vaccine manufacturers out for public use at some point. And the more that get approved, that may increase access earlier than expected.
Okay, that's a pretty good answer. Thanks, guys. So this from me, will this vaccine require yearly re-vaccinations?
I don't believe we know that yet. It depends. There's a potential that the vaccine could be potent for years and years. It's not really well known. The way you'd find out is you would do antibody tests and see what the antibody titer is in those 44,000 people Elsie talked about, or actually in the 18,000 or 22,000 that got the vaccine as well. Those are the ones you'd want to check, see what their titers are, and you could make decisions based on that. We don't know. We're flying the plane as we're building it, as Steve said.
Right. Right. I understand that. So from Jeff, KC1HB, Jeff Martin, one of your admins, to Ed or whoever, can you clarify? I think I missed something. You can get COVID again if you've had it once before, but once you get the vaccine, can you get it again?
Well, you can be exposed and you may even get infected, but you're not going to get sick. From what I understand, there are studies in England, and Elsie may know this better than I, or maybe Steve as well, that people who've been vaccinated, you can still culture virus from the nose of that individual, but they're not getting sick. That's why you still need to wear a mask. You still need to socially distance, because you may not get sick, but you may cough on your grandmother and she may get sick and die, or a child. And getting the vaccine and preventing that lock and key image that I gave you doesn't mean that the vaccine can't get into your nose again. It can. It just won't hurt you, supposedly, but it may hurt others. Does that help, Jeff?
Yeah, thank you. Helpful.
I think that was very well stated as well, Ed. That's exactly what I had read as well.
Okay, I'm on it. Give me a second. Thanks. Great answer. This next question from Robert Biancher, WB1GYZ from the group. A healthcare worker in COVID recovery with history of autoimmune and recent heart valve replacement is concerned about receiving the vaccine under mandate to remain employed in a healthcare facility. I'm not sure what the question is. That's what he played, but that's what he posted.
Well, part of it could be, is that a condition of employment? Getting a flu vaccine at Yale, New Haven is a condition of employment unless you have a religious or a medical reason not to take the vaccine for flu. There is no mandate right now for COVID-19. There are two questions there. One, does somebody with those many pre-existing conditions, would they benefit from getting the vaccine, or should they wait? And number two is, would the employer make it a requirement to, as a condition of employment, to be vaccinated? I don't know the answer. You would have to take it up with the HR department at the hospital that they work at. Medically, I would think if someone has that many pre-existing conditions, I think of getting vaccinated would probably be the wise thing to do. But whether it's a condition of employment is a legal question.
Bob, does that answer your question? Okay, I'll assume so. The next question from...
I tried to answer that, but pushing the space bar did not work. I see that I have to unmute. Yes, thank you for the information.
Thanks, Bob. Thanks for the question. The next question from Kristen Olson, KC1ISI. Ed, if they are finding the virus in wastewater, is there any indication the virus can spread fecal-oral route or through food from a symptomatic food service worker?
I would think, I don't know. I leave that up to, let me ask my public health expert here, Steve.
That's a great question. And, conducting food service inspections, we have not, to this point, again, I think we acknowledge that this is spread person to person. Some information that I have seen is that if the virus is on a surface, say a salad, that your stomach acid would neutralize that. But again, I think that there's limited information on that as well. But again, I think most of the information from CDC is it is spread person to person primarily.
Yeah, I have not heard anything about takeout orders. You'd think with all the takeout that's going on over the country, somebody must have COVID-19 while they're making your pastrami sandwich. Like we have the virus in addition to we have the meats. But it doesn't seem to be transmitted. So maybe it's killed by barbecue sauce. I don't know what it might be. It's hard to tell.
And conducting contact tracing, Ed, I can say that we have not gone back to an establishment that I'm aware of statewide that said, that led us to that conclusion.
Right. Usually it's the waiter breathing on you or coughing on you, or being in a room like, with Thanksgiving, you've got 12 or 13 people in a room with very poor ventilation. You're all breathing on each other. You're all taking your masks off if you're wearing them to eat. you're laughing, you're sneezing, you're coughing, you're speaking, you're singing, and the droplets are flying all over the place. You're much more likely to get that with your relatives in a house during a get together than you are walking down the aisle at the supermarket where somebody may be infected, but you're walking past him and you're just in close contact with that individual for maybe 2 or 3 seconds. You're not going to get enough virus from that contact, whereas you do get it from being, at large gatherings. So it's all about the ventilation, which is why being outdoors was helpful. Also, the ultraviolet rays of the sun inactivates the virus. The worst thing that could have happened was for us to go indoors in the peak and then have two holidays back to back in a devastated economy. It's really if we were writing a doomsday scenario, this would have been it.
And the virus in and of itself being SARS-CoV-2, being an upper respiratory illness, would lead me to believe that it would be person-to-person transmission as opposed to, say, norovirus, which is known as the cruise ship virus. That's typically obtained by touching surfaces and consuming food. So that I think there's a difference there.
Yeah, and I don't know about the fecal-oral route, Kristen. That's an interesting question. I don't know why it's not transmitted by blood. It's not transmitted by blood transfusion, which is very helpful. Otherwise, we wouldn't be having a lot of surgeries anymore because they need blood to do the surgeries around the country. It doesn't seem to be bloodborne.
Okay, so I'll ask, is Kristen, does that answer your question?
Yeah, no, that's information I kind of already knew, but I was interested in the wastewater aspect of it. So that's why I didn't know if there'd been any research or changes to that. So that does help. Thank you.
Okay. All right, let me get to the right spot.
Not human related as well, but I know that there's been some discussion about how that impacts the ecology around it. If there's wastewater that's, not treated properly or in the treatment process, you may not treat for all that virus. You are seeing different animals start to test positive in nature. And that's something that we've seen more of recently.
Way over my pay grade. So the next question is from Tiffany to, let's see, aspirating is no longer necessary or evidence-based for many intramuscular injections. You are right. There are not many blood vessels. It sounds like a statement. Is there a question there?
Yeah, no, she was just making a statement.
I was just answering the question about aspirating and that you weren't seeing that in the video.
So what was the rest of the question? So the answer was what? It's not because there are not a lot of blood vessels, we're not seeing aspiration?
Right, and I also just included below the CDC recommendations for IM injections don't recommend aspirating anymore.
Oh, well, that's what happens when you live under a rock. You miss all that good stuff. Thank you very much. I appreciate that. Thank you.
No worries.
Okay, thanks. The next question, Katie Asmus. Can you clarify? This vaccine attaches to the spikes specified to the corona vaccine, or will this vaccine work against other RNA virus, such as Hep A, Zika, flu, etc.?
Well, my understanding is I don't know enough of microbiology or molecular biology to know if those other viruses, and there are different kinds of viruses. Zika virus, I think, is a flavivirus, and this is an RNA virus. I think they would have to, the antibodies against the spike protein. So unless those other viruses had a spike protein, even if they have a spike, the protein in that physical spike would need to be what the antibody is looking for. It's quite specific. Each key has its fits a separate lock. So there would have to be a virus that had the same protein in it that the antibody was looking for. Otherwise, it would not be effective. Which is why the mention of antivirals would be interesting, because that's generally not specific for a part of the virus, but it may be something more viruses would have in general, or it would hit an enzyme that might affect multiple viruses. So it would be less specific, but in this particular case, it would have to be right at that protein site.
Thanks, Doc. Katie Asmus, does that answer your question?
Yes, thank you.
Perfect. This next question from Tiffany from the CDC. Aspiration before injection of a vaccines or toxoids. In example, pulling back on the syringe plunger after needle insertion but before injection is not necessary because no large blood vessels are present at the recommended injection sites, and a process that includes aspiration might be more painful for infants.
You just answered that.
I thought so, but I'm posting the question, guys.
Okay, well, thank you.
I'm just a guy here. Okay.
Everyone's got to have a guy. All right.
Yeah, I got a guy. You know, I'm a guy, so that's what happens. Moderna versus Pfizer from Jeff. One more effective than the other?
That was from you.
It is from me. Sorry.
My understanding is they're equally effective from the clinical trials. What'll
Why
happen
did-
when it gets to millions of people, I don't know. Elsie, any thoughts?
Same answer. Right now, they look the same.
So
We don't
why?
have any more data yet.
Okay, so the reason for the question is, why does one require such a lower degree of temperature than the other?
It's the way it's made, and that's proprietary. Yeah,
Okay, I get that.
it's kind of where we are.
No,
I get that.
Et cetera, et cetera.
That's also above your pay grade, Rob.
That's above my pay grade. You're right, Jeff.
Mine too.
By the way, the blood banks, which I run, we've been storing frozen red cells at -70 for 50 years. It's not a problem getting down to -70. -30, we store plasma in constantly. It's not a problem. The problem is shipping it. And when it gets to a site, it doesn't sit around on a loading dock for 2 days till somebody gets to it. Or it gets put in a refrigerator that's not up to the right temperature. It's not that hard to ship something at -70. I mean, it's harder to ship it than it is to actually have it in a fixed site.
Right.
-30 is easier, but I don't know. -70, I think, still runs on 115, but it may be 220. I don't know. Is that 220 or is it 115? Volts.
No, it's all probably 120, 115.
Yeah. Okay.
All right, so this is from Bill W1KKF. Bill, you're going to have to unmute yourself for your question. He has a question versus concerning transmission. Go ahead, Bill.
Hi.
Okay, actually, Linda has the question. Go ahead.
Yeah, I've been nominated. We see our 11-year-old grandson for a few hours on most Wednesdays. He is going to in-person schooling 4.5 days a week. His mother volunteers 1 day a week doing bookkeeping at the food bank here in Wallingford. His father goes to work every day. I believe he has his own office. And Bill is a little bit uncomfortable about hugging him. We wear masks, we turn our faces in opposite directions, but Bill's concerned about transmission on clothing. What's the chance of that?
Well, I would agree with Bill for the first time in many years. I don't think hugging is appropriate. Not so much because of the clothing, which could be, but there's facial contact. There's really no need to hug somebody. We joke about hitting elbows and things. It's really the way to go. Everyone's wearing a mask and you're protecting yourself. And there's one chink in the armor there where you get too close to someone. There's really not a need to do that. You can express love in my own thoughts in other ways. So I agree with Bill. It's not as good a practice as you would like it, as you think it might be. Other comments?
Okay, no, that's it. Thank you.
That's it, Bill. I'm not going to agree with you again for another 10 years.
I don't agree with him very often either.
Okay, so next question. And this is from John Bart Shearer. Sorry to ask for a repeat. I missed the answer to the question about the shingles vaccine. I'm in the same situation.
Steve, did you want to take that?
I think that was, did you handle that one? I think to the effect of that. I think you handled that one.
All right, I wouldn't take one on top of the other, but if you're getting a shingles vaccine, I do not believe it's a live virus, so I don't think it's, what you want to do is you don't want your body to be hit with so many foreign proteins that you're not going to get as robust a response to them if they were separated. So I would think a couple of weeks or maybe a month separation. I mean, you're not going to probably be able to get the COVID vaccine until sometime in the summer. So if you have a month or so between your shingles shot and your COVID shot, you should be fine. That's my thought, but check with your provider, and I think that's the right answer, though.
Okay, John, does that answer your question?
Nay.
Nay, but yes, thank you, Ed. I'm sorry to have to ask you to repeat.
That's fine.
Okay, effectively, that's the end of our questions that I have in my list. I'm going to open up to anyone that has not posed a question in the chat. Anybody have a question for the panel before we end the membership or end the meeting here?
Yes, I do. W1EDX.
W1EDX did not pose a question, but go ahead.
This is being recorded, right?
Of course.
Yeah, apparently it
Okay,
is.
so it's going to be, I sent an email out already. So it's going to be available to view on YouTube.
No, I'm not guaranteeing it's going to be available on YouTube.
So how would one look at it again?
You're going to have to ask me, Paul.
For a small fee, we have to give you a private showing.
No, seriously.
No, I am serious. I have a day job. I'm very busy. I've got a family of 6 in here in this house. I've got priorities I have to maintain. So I will make it available, but it may not be very soon.
Oh, okay. No, I was just wondering if it was going to be available like YouTube so other people can view it.
I would love to be able to do that instantly. I just can't do it.
Okay. Fair enough. Thank you. Have a good day.
Sure, buddy. Anybody else with any questions for the group?
Yeah, Rob, you missed my second question. I thought I sent it in.
I didn't see it. I just went through numerically. Go ahead.
Okay. Yeah, I saw something on television that they are now training dogs to detect COVID, and I was wondering what you folks thought about that.
They have dogs that can detect cancer in patients, which is amazing. They secrete, I'm sure, certain chemicals that give off an odor that dogs can detect, and I'm not sure what they call them, but they're dogs that can sniff individuals and determine if there's a malignancy somewhere. So I'm not surprised that they have something for COVID as well. We humans think we're really smart, but I think animals in many ways are smarter than we are.
Animals are much more instinctual than we are.
Yeah.
Ed, I have a
probably right.
quick question for you, Ed, if you can clarify something or just add something on. So earlier you were talking about people who had allergic reactions to like peanuts and stuff. And is that related to what I read in the news about people with Bell's palsy or like their face being paralyzed from taking the vaccine?
No, when you get swollen up and your face gets all swollen from peanut allergy, it's your.
That's a bad thing.
The cells, the cells are very close together, but when you get these anaphylactic type reactions, the cells move apart and liquid leaks into it, so you get fluid moving between cells and normally it doesn't. That's why your face gets all swollen. goes away over time. Bell's palsy is a damage of part of the eighth nerve that can happen with viruses. Often herpes can cause that. That usually recovers to a large degree, but some people may be left with residual activity. That's a different issue. You don't get Bell's palsy that I know. It might be. I never thought of getting Bell's palsy from a severe allergic reaction. I don't believe that's. I don't.
Well, I want to say I read in the news this morning that people were getting that from this vaccine.
Bell's palsy?
Yes,
They're,
I
yeah, they were
heard the
getting
same
like
thing.
facial paralysis.
I heard the same thing.
Did you? That I hadn't heard that. I would have to look into it. That would be not so wonderful if that were true. I don't know why 44,000 people would. I don't know how many vaccines they've given out so far, but we're hearing all kinds of reactions that were not in the clinical trial. But there was a drug fen-phen, which I think was to be used for weight reduction. Turned out that it caused damage to heart valves, which wasn't predicted. And they had other vaccines that caused intestinal problems. So I'll have to look into that. I don't know anything about that.
Sure.
Maybe right, and I stand corrected.
I think you need to use or pick your poison. Any other questions for the panel?
Yeah, this is Mike. I have a quick question.
Go ahead, Mike.
Now, I stopped working as a driving instructor because of the COVID virus. Since I stopped working, some of the driving schools have gone back to having driving lessons. But because of the medical history I have and my age, I don't want to see myself in a position where I'm going to be 2 feet from a student who probably isn't wearing a mask around their friends. The question I have is, if I get the virus and I'm wearing a mask, would it be safe for me to be working in that environment, or should I just forget it altogether?
I would not recommend being in a closed car with all these different people unless I was vaccinated and you've had both shots, and then you might want to consider it still wearing a mask. You're very vulnerable, Mike, and being in a car with the windows up, even if you're wearing a mask, is not a good idea because you're just both rebreathing the same air. So until you're vaccinated, and I would think that's not a good idea. That's my own thought, but obviously check with your other doctors.
Well, just to clarify, my question was based on after the vaccine. If after I get both vaccines, if I were to wear a 95 mask, for example, would it be safe for me to go back and work in that environment?
Well, if you're vaccinated and you have antibody, I would have to say yes, it is safer. But I would actually want to talk to somebody else about that. I'll check with some other people that I know and get back to you. And we can get everybody together again and they can hear the answer. So I'll let you know. I'll let you know. If I think the vaccine works, I'd have to say, yeah, it would be fine. But I hadn't thought about that, so I'll get back to you.
Okay, is there anyone that has any other question for the panel? No other questions, panel. You are disposed.
All right, well, let me thank everyone for coming in. We had up to 56 people, I think.
We had up to 63 at one point.
We did. Oh, that's even better. I'd like to thank Steve Civitelli and Elsie Mathews for joining me. Thank our administrators, Jeff and Rob. And thank you for checking in and listening. Hopefully you found this helpful. You found it non-judgmental. We tried to be as forthright as we could. And if you have any additional questions that you would like some answers to, if you want to get in touch with me, it's w1ysm@arrl.net, and I'll try to get an answer for you. This was just an example of how our Meriden Club, working with the Department of Public Health in Wallingford, tries to go beyond just amateur radio, but the community service, and we really appreciate the cooperation and everyone's participation. So thank you all. Stay well, mask up, stay apart, and stay safe. Thank you.
Superlative effort to the panel. Thank you for appearing and everybody who contributed.
Thank you, everyone.
Thank you.
Thank you.
Stay safe.
Yeah, thank you.
Yeah, you're welcome.
Stay well.
Thank you. Well job, well done.
Thanks,
Good night.
Jeff. Thanks, panel.
Okay, good night.
And I
all.
just looked up Bell's palsy, and it said brief facial palsy was seen that was transient. So I'll have to look into it further.
Can I tell you? I hope it was the other palsy where you throw swear words at people. So when I get my vaccine, I can legally swear at people and they can't hold it against me.
That's Tourette's syndrome. That's completely different. I'm sorry.
Tourette's, yeah. Well, that's what I was hoping for.
I don't give
You
a
guys
damn
know
what
me
the
by now, and that's what I was looking for.
I was going to mention, Ed, Jeff K1VG put a link in the chat, and apparently the Bell's palsy is unrelated.
Oh yeah, okay.
That's what the link said.
I heard that outside of this group. I've heard that outside of this group, so that's why I guess I brought it up.
Okay, I would think if it was with the vaccine that you'd see it, you know, with the illness as well. I don't know. I'll have to look into that.
Sure.
Okay.
Good
Thank
luck with
you,
the snow, people.
Thank you very much, Rob and Jeff.
What are
Appreciate
you
it.
doing?
Jeff,
Yeah,
thank you
it's
so
fine.
much for chipping in here, buddy.
It's
Oh,
on.
of course. Yeah,
Yeah.
my pleasure.
Thanks, administrators.
Thanks, Elsie.
Okay.
Take care.
Well done.
Yeah.
Thanks, Elsie, Ed, and Steve.
Bothering me. Don't forget
Good
to
night, everyone. I'm out of here.
Yeah,
Night.
me too.
Night.
Good night, all.
Night.
Sayonara.
Night.
Great job, guys.
Thank you. Take care. Bye-bye.
Bye-bye.
Yeah.